So now I'm going to introduce a few illustrative examples of work that I've been doing from the Cochrane Epilepsy Group. And this is an IPD network meta-analysis in epilepsy, which considers 10 different anti-epileptic drugs used in monotherapy, so single drug treatment. So these are the drugs that an individual would receive when they have just been diagnosed with epileptic seizures. So just to briefly talk through the network diagram here on this slide, the dots, the nodes, the blue dots show how many studies a drug has actually been included in. So we can see the most commonly used drugs such as CBZ, carbamazepine, BPA, sodium valproate, and LTG, PHT are also very commonly used drugs. And the lines and the thickness of the lines show how many patients have been included in the head-to-head comparisons of those drugs. So, for example, we can see a lot of patients have been included in trials comparing CBZ and LTG, also CBZ and VPA. And one of the benefits of using a network mesh analysis for this example is that we can also make indirect comparisons. So, for example, if we look over to the left-hand side at PB and OXC, there is no line in between those two drugs, as those two drugs have never been compared head-to-head within a trial. But using network meta-analysis techniques and indirect comparisons, we can calculate a relative effect size to see how those drugs would compare head-to-head in a trial without actually having to conduct the trial. So for our 10 different drugs, we can make 45 different pairwise comparisons of interest via direct and indirect evidence. And we also have some related pairwise Cochrane IPD reviews of anti-epileptic drug monotherapy. I'll just briefly flick through these slides now, showing links to these reviews. So we've got lamotrigine versus carbamazepine, also topiramate versus carbamazepine. Most of these reviews do involve carbamazepine as it's a first-line treatment. Carbamazepine versus sodium valproate, carbamazepine versus phenobarbital, oxcarbazepine versus carbamazepine, carbamazepine versus phenytoin. And we also have other reviews involving phenytoin, which is a commonly used drug in the States. So phenobarbital versus phenytoin, oxcarbazepine versus phenytoin, and sodium valproate versus phenytoin. Okay, so for these epilepsy reviews, why did we use an IPD approach? So going back to the questions that I posed in the earlier slide, the outcomes of interest, firstly I'll start with, for these reviews, are time to event outcomes. So the primary outcome is time to withdrawal of allocated treatment, and the secondary outcomes are time to first seizure recurrence and also time to 6 and 12-month remission of seizures. So we have time to event outcomes here, and it's well documented that time to event outcomes are not very well reported, certainly within oncology. So would the summary level data that we require for an aggregate data approach be reported in these epilepsy studies? Also, further considering our primary outcome of time to withdrawal of allocated treatment, this outcome actually has a complex definition, as there are many reasons that an individual may withdraw from their treatment. Perhaps it's not working for them, they're still having seizures, perhaps they're having adverse events, or there may be other reasons why someone has stopped taking their treatment. But really what we're interested in is whether the reason for withdrawal is treatment related or not. So a drug not working, a drug causing side effects, we would consider to be an event within our analysis, whereas other reasons such as loss to follow-up, non-treatment related death, we would consider to be censored in our analysis. However, the way that we are defining this outcome and the way that the individual studies have been defining this outcome have been quite variable. So this outcome is actually defined quite inconsistently across the literature. So even if the summary level data that we need is reported within the published literature, should we really be combining data for this outcome when the definitions of the outcome are inconsistently reported? That might introduce heterogeneity into our meta-analysis. So also considering our clinical question and the objective of these reviews, we're also interested in measuring the relationship between treatment effect and a covariate. So here we're interested in epilepsy type. As NICE in the UK, and I believe that other guideline developers globally were making similar recommendations at the time. So NICE was recommending in 2012 different first-line treatments in monotherapy for people with so focal onset seizures, which is the most common epilepsy type that around 60% of people have. The recommended first-line treatments for this seizure type are CBZ and LTG, whereas for individuals with generalized onset seizures, so the less common seizure type that around 40% of people have, BPA, sodium valproate, is the more commonly used treatment in this recommended treatment for these people with this seizure type. So in our pairwise meta-analyses, all of the analyses are stratified by epilepsy type, and within the network meta-analysis, we intended to incorporate a treatment covariate interaction to allow us to estimate separate treatment effects for all the different 45 pairwise comparisons for each of the epilepsy types. So to summarise for our epilepsy reviews, our clinical question, our objective, there is an interest in modelling treatment covariate interactions, i.e. doing something complex. Also, our outcomes of interest are complex time to event outcomes, which are not consistently reported, and the time to event statistics also are not often reported within the published literature. So in our example, we deemed the IPD approach to be the most feasible and the most sensible approach for this example. So these are some questions, some thoughts that you could consider when deciding whether you should be using IPD or aggregate data in your particular review for your clinical question. So now that we've decided that we are using IPD, we need to request some data. So I'm just going to go back in time, have a little history lesson here, thinking about the early days of IPD meta-analysis. So the first IPD meta-analysis that I can find published was published in 1987. And before the year 2000, in the early years when this was a fairly novel technique, I personally wasn't involved at this stage, but from speaking to the team that I work with, some of whom were involved pre-2000 in IPD meta-analysis, the methods of requesting data at this time, you would have to write a letter to a researcher who had conducted a trial, maybe pick up the phone and speak to them to request their data, or have a face-to-face meeting with lots of collaborators to perform an IPD meta-analysis. Often data was provided on paper, maybe sent in the mail, sent by fax, and then would have to be computerized by a team to be able to conduct analysis. And also generally in this time, everyone within the clinical area knew everybody else conducting studies. So data sharing was very informal. Friends shared data with their friends without too many questions being asked about what was being done with that data. Now coming forward in time to post-2000, and then I got involved in this sort of work around 2011. Data requesting did come into the digital age, so around the time that I started doing this, all the requests were being made by email, and data was also being sent by email or on CD, sometimes on floppy disk. And also around this time, new research groups became established across the world. So certainly within epilepsy, for the first time, new research groups were being established in developing countries where they hadn't been based previously. So it was no longer a case of everyone knowing everybody else, and a lot more questions started to be asked about, why do you want my data? What are you doing with my data? So the late 2000s saw the introduction of the first legal documents for IPD meta-analysis, data sharing agreements, outlining the role of those providing data, the role of those requesting data, what can and cannot be done with the data, what can be published as results, and so on, just to protect everybody involved so everyone knows what can and cannot be done with that data. And then thinking about where we are today and the present day of data requesting and where we're heading in the future, we are now in an era of data sharing platforms. So I've highlighted a few of the largest data sharing platforms here: CSDR, clinicalstudyDataRequest.com, the Yoda project based out of Yale University, and also Vivly, which is the newest data sharing platform, which was launched this summer. So data sharing platforms have existed since 2013, mainly being used as a platform to host pharmaceutical data, but more recently in the last few months, some academic data providers have also made their data available on these platforms. And also since 2015, there's been a lot of data sharing policies, data transparency and data sharing policies made public by some very big names in the academic community, such as the Institute of Medicine, the European Medicines Agency, the International Committee of Medical Journal Editors, and also surveys of trialists and patients since 2011, compared to earlier surveys, are now showing support of clinical trial data sharing, that this is a positive thing that we should be doing to bring science forward. And really the recent years have marked an era of data transparency across the research community as a whole. Now attitudes are very positive towards data sharing, that this is definitely a thing we should be doing. So thinking specifically now about our Cochrane Epilepsy IPD reviews and requesting data for these, another step back in time, as our team has previously published an IPD network meta-analysis of epilepsy monotherapy trials, which was published in 2007. So for this project, which was requesting data from randomized control trials, which compared 2 or more of 8 anti-epileptic drugs, and the data was requested from these studies between around 1995 and 2005. And the team managed to get data from 19 out of 30 trials, so 63% of the total trials. And out of these trials, they did manage to get the majority of the patient data, so 4,703 out of 5,887 participants, so 80% of the participants, which I do consider to be a very good retrieval rate, especially considering as some of these trials were requested back in the days where writing a letter, receiving data by fax would have been the method of receiving and requesting data. So also adding in IPD from 2 trials that were conducted at the University of Liverpool with about 2,500 patients, bringing the total number of patients up to over 7,000. And this work was published in 2007. Then plans to update this as a Cochrane IPD review with newly published evidence and also including 2 newly licensed drugs. The plans were started between 2010 and 2011. And a systematic search was conducted in which 45 new trials were identified from journal articles and published abstracts. So the data requesting started in January 2012 for these 45 new trials. And how did we go about requesting data from these trials? We wrote a short invite letter and sent a data request form to the corresponding authors of all of the journal articles that we'd identified by both email and by physical mail. And in the event of no response, other authors of that journal article with a traceable email address were contacted. We didn't specify any particular format that we wanted the data to be in. We would accept any format, and there was no specific deadline set, as we wanted to make providing data as easy as possible for these academic groups who were likely to be very busy, and we were willing to do all of the work in preparing the data. So what happened? Well, progress was very slow. We met some obstacles in working out who owned the data and who could provide access to the data, as often we received replies from academic authors stating that the pharmaceutical company who sponsored the study owns the data, only they can provide access to it, and at the time it was very difficult to make contact with pharmaceutical companies for research requests. So by the end of 2013, after 2 years of data requesting, IPD from only 2 publicly funded trials was available to us, which was 3% of the total data. So it wasn't looking very positive after 2 years, and we'd be able to update this work anytime soon. And then in January 2014, a landmark date where the first collaborative data sharing platform, CSDR, was launched, which was initially a collaboration of GSK and Roche. And also throughout 2014, other pharmaceutical companies were developing their data sharing policies and putting together data transparency teams. So it was now much easier to make contact with pharmaceutical companies to request data. So we made our requests again for the 16 pharmaceutical sponsored trials of interest to us. 6 of those requests were made directly from clinicalstudyDataRequest.com, and 10 trials were requested directly from the sponsors, as these particular pharmaceutical sponsors were not part of the data sharing platform at the time of the request, but all of them are now. And also, due to this renewed interest in data sharing across the entire research community, we decided to try and capitalise on this and made all of our outstanding requests to 27 publicly funded or government funded studies again. So how does one go about requesting IPD via a data sharing platform? Well, the sponsors provide a list of studies which are listed by their sponsor ID or their NCT number, which may be available on clinicaltrials.gov. And those are the studies where the IPD is available to request. However, if the study isn't listed, you can submit an inquiry to check the availability of the IPD for that study. And a feasibility check is done by the trial sponsors to check whether the IPD could feasibly be provided for that study. Then once it's been established that there are studies there and available which are of interest, the team submits a scientific research proposal and details of all the research team members, and at least one team member must be a qualified statistician. And also the research proposals must include some kind of publication plan. And the research proposals go to an independent review board for approval based on their scientific merit, and some modifications or clarifications may be requested by the independent review board prior to approval. Following approval, a data sharing agreement is signed by both the sponsor and the team requesting the data, and then anonymized data is provided within a remote analysis platform, where statistical software is available within the platform so that analysis can be performed, and then the results can be exported from the platform. But in most cases, the data cannot be taken out of the platform. The data remains there. It's only the results that can be taken out. But there are now a few sponsors which do allow their data to be downloaded temporarily from the platform. So for us, what happened next? Well, we stopped our data requesting in December 2016 after around 5 years of requesting. And by that time, by the end of 2016, our IPD retrieval rate was 15 out of 45 trials. So we got data from a third of the trials, but we did get data from over half of the participants. So 5251 out of nearly 10,000 participants. And one of those trials with 136 participants, the IPD was provided to us in the remote analysis platform. And then adding in this new evidence from the data that we previously had, that then brought us up to data for 12,391 out of 17,961 eligible participants, so 69% of the total data from 36 out of the 77 eligible trials. So we had data for over 12,000 participants from 36 trials, which were published over a 30-year period. These trials were in different formats, different languages, data being collected in different ways, and was really of very different levels of quality. So we did require a standardized approach to preparing this data for analysis. And our first step was to perform checks of the IPD we'd received according to any published reports in the published literature. So just checking that we could reproduce any results, that the numbers of patients that we had agreed with published literature and so on. And any issues that we had, inconsistencies or uncertainties, we discussed with whoever provided the data to us. And then once any inconsistencies had been resolved, or if there weren't any inconsistencies in the first place, we calculated our primary and secondary outcomes. And 2 statisticians independently did all of this. So we didn't accurately measure the amount of time it took to prepare each data set, as we didn't consider it to be a fair comparison when we had trials published over such a different time period, different sizes, different methods of collecting data. And we also weren't racing each other as statisticians. We had more interest in the data being prepared correctly than being prepared quickly. However, to make an observation, as I looked at all of these data sets, the data provided from 2014 onwards, particularly the industry-sponsored data, did seem to require much fewer checks and very few, if any, uncertainties or inconsistencies, which allowed us to prepare data for analysis relatively quickly. So I did perceive there to be a higher quality of the recent pharmaceutical data that we received, which was very likely to be reflective of the extra attention that was being given to these data sets by the data transparency teams, the teams performing anonymization prior to sharing. So as I mentioned, we did have one trial with 136 participants provided to us in the remote analysis platform. And unfortunately, we had to exclude that study from our IPD analysis, as we could not download the data at the time. And even though other data sets can be uploaded into remote analysis platforms, some of the data sharing agreements that we had requiring us to keep our data in a secure network that was under our control, that prevented us from uploading the data to the data sharing platforms which were not within our control. However, this was only 1% of our overall data, so it's unlikely to have influenced our overall results. But we did get data for 7 other industry-sponsored trials, the data was provided to us directly, as these sponsors were not part of any data sharing platform at the time of the request. However, now, today, all of these sponsors are now part of a data sharing platform. So for 4 of these trials, the data would have been provided by CSDR. For 2 of them, it would have been via the Yoda project, and 1 would have been provided via Vivli. So that's 7 trials across 3 different platforms. And in those trials, we had 4,534 participants, so 37% of our total data. So if we are not able to get all of this data together to be able to perform our analyses, that's a much larger amount to potentially exclude if we cannot combine it. So this is something which is of concern looking forward for IPD synthesis. So now just to briefly summarise the results of the Cochrane Individual Participant Data Network Meta-analysis, which was published at the end of 2017. And so I noted that the planning of this project started in 2010, and it wasn't published until 7 years later. This is how long these projects take. So to briefly summarise the results. So for individuals with focal onset seizures, our results support the NICE guidelines that CBZ and LTG are suitable first-line treatments. And our results also show that one of the newer drugs, LEV, may also be a good alternative for people with focal seizures. For people with generalised onset seizures, the results also support NICE guidelines that VPA is the most effective treatment. However, recent work has shown that VPA sodium valproate is associated with birth defects, both physical and neurodevelopmental, so this drug is now no longer recommended for women of childbearing potential. And therefore our results have shown that LTG and LEV may be good alternatives for those who cannot take VPA, for people who have generalised onset seizures. We must remember that these results are based on 69% of our eligible data, and that 31% of the data is missing from these results. And what should we do about that? Perhaps we should just ignore it and hope no one notices, interpret our results as if we did get 100% of IPD. And this is unfortunately what 25% of published IPD meta-analyses do. They make no reference to the fact that they didn't get all of their data. But we must consider the risk of availability bias, in that the available data may not necessarily be reflective of the entire evidence base. So what if the trials that haven't provided IPD are different to those that have provided IPD in their characteristics and their results? Perhaps we can examine whether any aggregate data is available for these trials not providing IPD. Perhaps the reasons that IPD is not available could be informative, could be related to the results. And overall, this means what if our results and conclusions are actually biased by this availability bias? So considering the first question, are the trials without IPD different to those providing IPD? So we can consider trial characteristics, results such as demographic, sample size, publication year, funding, centres, bias, positive, negative results, and so on. And looking at the epilepsy work, where we had 36 trials with IPD and 41 without, from an examination of the characteristics and results, we found that the provision of IPD does seem to be associated with the resources of the trials. So the large multi-centre pharmaceutical trials seem to be providing more IPD than the smaller single-centre trials that are maybe conducted within academic institutions. And it was difficult for us to assess whether there was any impact from the positive or negative results, as the majority of the trials didn't actually report any statistics for the time to event outcomes of interest to us. So that was a difficult assessment for us to make. Considering the reasons why IPD were not available, if any reasons were given to us, and if the reason for unavailability could be related to the study results. So thinking about the two different time periods of data requesting, so for the first project the team did, and then also for this updated work, throughout this the whole time frame, some of the reasons quoted were that data had been lost and destroyed because the ethics had expired and the data wasn't recorded in the first place, or in quite a few cases it wasn't possible to make contact with the necessary person to request the data. Then also in the more recent requestings for the Cochrane IPD work, some of the reasons that we got were that data could not be anonymised, data sharing was not permitted by the ethics or the patient consent forms, and then again also thinking back to the previous slide, but the resources, the costs of providing data were prohibitive. So here again we've got another poll, another question here, just thinking about these different reasons that we've got. Could these reasons be considered informative? So we've got some responses for you, so have a think about this and then cast your vote on the poll. Okay, so again we've got some mixed responses here. So the majority think yes, these reasons are informative, 44%. We have 37% with I'm not sure, and 19% think no. Well, in some ways this is a bit of a trick question, as I don't think there is any correct answer to this. It's very subjective. I personally wouldn't consider data not being able to be anonymised or data sharing not being permitted by original ethics to be informative, but perhaps costs of providing data being prohibitive, so the resources of a trial being related to whether IPD is available or not certainly could be informative. Not necessarily related to the study results, but yes, this certainly could be an informative reason, and we should bear this in mind when we are interpreting our results. Okay, so thinking about now whether the results and conclusions may be biased due to this 31% of data this is missing, we can explore this availability bias visually and statistically. So this is a comparison adjusted network funnel plot. So this is an extension for network meta-analysis of a classic funnel plot that would be used to examine publication bias in a traditional network meta-analysis. So again, just another quick poll for those of you who are maybe familiar with looking at funnel plots. From the information here, do we think that any publication bias or availability bias could be present? So here we have another poll of yes, no, and not sure. Okay, so the majority of you think no, there is no publication or availability bias here. 24% think yes, and 21% are not sure. Okay, so I'm just going to move on to the next slide just to discuss this a little bit further. So here you would consider the spread of the dots on this plot in the same way as you would for a traditional funnel plot, looking for any asymmetry, and particularly if there are any maybe small study effects missing. And also here we have a line which is the equivalent of Egger's test in a classic funnel plot. We're testing whether the gradient of this line is different to 0, which would indicate publication bias. So a significant p-value here would suggest significant asymmetry. In other words, there may be publication bias. So from that test, and also just looking in general at the spread of those points across the plot, I'm not particularly concerned that there's any evidence of publication or availability bias here. So this is quite an encouraging plot, certainly for our primary outcome, and we could also consider our secondary outcomes as well. Okay, so thinking about these 31% of data that's missing and the studies that these come from, is there any potentially aggregate data, summary statistics from these trials without IPD, as there are methods out there for combining IPD and aggregate data in meta-analysis that we may be able to use? So actually, for these epilepsy trials, the majority of them didn't report any of the time to event statistics for our outcomes of interest. There was some summary level data available for 10 trials without IPD out of the 41. However, we still did have concerns over inconsistent definitions of our primary outcomes, so maybe we didn't actually want to combine this information with the data that we'd standardized. And we did have to treat that one trial in the remote analysis platform that we had to exclude from our IPD analyses as aggregate data. But actually, the main problem for us was our interest in the treatment effect by epilepsy type, and there was very little aggregate data available separated by this covariate. So we were only able to add in an additional 2% of data for people with focal seizures and an additional 0.7% of data for people with generalized seizures, so a very small amount. And unsurprisingly, this additional small amount of data had a negligible impact on our primary analysis and our conclusions. So aggregate data really wasn't useful to us in this particular example. So taking into account all those different questions that I posed and thinking about whether our IPD network meta-analysis could be at risk of availability bias. So are the trials different that have IPD compared to those that don't? Are the reasons informative? Well, in both of these cases, we seem to be finding a recurrent feature that the provision of IPD does seem to be related to the resources of the trial, which is concerning for our results. It's difficult for us to know whether the results of those trials that didn't provide IPD were different to those that did, as there was very little aggregate data available. We didn't have any statistical evidence from our funnel plots of availability bias, which is encouraging. So in conclusion, we don't think that the 31% of the missing IPD has had a large impact on our results, but we should interpret our results appropriately with availability bias in mind and certainly not ignore it.